Well, AAIC is the global conference on Alzheimer's disease. It's an international conference on Alzheimer's disease, and all Alzheimer researchers from all over the world convene to discuss their latest findings. So this year, I expect it to be very special because there are a couple of things that are to be announced. For example, there will be results of successful trials targeting Alzheimer's disease, and that will leverage a very positive vibe. There are other results that will also be shown. For example, there is a panel discussion on the novel criteria to diagnose Alzheimer's disease, and that's also very important and exciting. So the difference from other neuroscience conferences is that the focus is on Alzheimer's disease and that it's really a mix of basic Alzheimer science and research, and also clinical research. So I really like it also it's every year a nice happening, and also seeing friends and other researchers again, and enjoy together how nice it is to perform science. So you asked me a lot of questions. So key challenges for early detection, for also early prevention, and for treatment development. So for a long time, it has been difficult to address the Alzheimer's disease because we didn't have a clear diagnosis, and the brain is really not accessible to look into. So it's you cannot make a biopsy like in cancer. So we have to do with alternative strategies. And luckily, there are very good options, for example, to look into the cerebrospinal fluids, the brain fluids that immerses the brain. But also imaging methods are very helpful to make a diagnosis during life, because the gold standard is neuropathological research, but that's an investigation that you can do only post-mortem. And and we want to know what's happening at the time of diagnosis, which can be decades before the patient passes away, and we can make an inspection of the brain. So, and for a long time, for these researchers, studies into the cerebrospinal fluids, we could only investigate the biomarkers that are related to the pathology, and it's always difficult to make novel methods. But nowadays, there are very sensitive methods that we can measure also the Alzheimer proteins, exactly the core proteins of Alzheimer's disease that we used to measure in CSF, cerebrospinal fluids. We can now measure them also very reliably in blood. So there's opening a novel area due to technical improvements. And of course, we need such biomarkers and special blood tests. They will be very helpful for studying effects of prevention, for example. If you have a lifestyle change and there is a change in your blood biomarker, hopefully that relates to each other and relates to reduction in the risk of Alzheimer's disease. And also for treatments, we are reliant on biomarkers in body fluids, also because the cognitive tests are usually not so sensitive towards changes, and there are various cognitive tests. They all show more or less something different. So they can be very noisy also, depending on the background or the way that questions are being asked. And if you have a biological marker, an objective test that you can measure with a kit, and leads to a numerical result, then we have a more reliable or more robust outcome measure. So nowadays, we think that with the pace of development of the Alzheimer biomarkers and outcome measures, we have a good basis to define Alzheimer's disease and also to use such biomarkers in a treatment evaluation. So there have been a tremendous technological advancement in the field that will help the development of novel biomarkers for Alzheimer's disease. For example, nowadays we have ultra-sensitive technologies, several microfluidic technologies, but also novel array-based proteomics technologies like Olink. That's very nice technology if you think of bringing an assay to the clinics, because in the clinics we use immunoassays, and immunoassays, they are composed of two antibodies in a sandwich format to detect a protein. And in the past, we were reliant on mass spectrometry analysis, and that's a really powerful tool also for identification of novel mechanisms and novel proteins that are relevant. But we were not so successful in translating those findings, proteomics findings obtained by mass spectrometry, to clinically useful tests. And nowadays, with, for example, an array like Olink, we have already proof of concept that there is an antibody pair, and that's the basis of the immunoassays in the clinical practice. So when we do a discovery of a protein in the Olink panels, we know that there is an antibody pair and that we can use for further downstream development towards a single assay or a kit with a couple of biomarkers, so multiple assay up to 10 or maybe 20, but we don't need 1,000 proteins anymore. And that will give us a lot of information that we can use in clinical practice. So the Coral Consortium is a consortium that's composed of researchers that are interested in collaborating together using Olink proteomics for neurological diseases. And by establishing this consortium, we want to reach the aim of improving the research and the identification and the validation of novel biomarkers that are potential in the neurological space. So, we organize seminars for exchange of experience, workshops also for exchange of experience, and for example, we do meta-analysis together. So that is really leveraging the available knowledge about using the Olink proteomics. And also actually leveraging the availability of samples that we can exchange, but also the data sets, because it's very unique that all use the same Olink approach. So it's the same technology, and that allows you to compare the results between different studies. So that's really what makes it unique. It's not just a collaboration into biomarker researchers. It's biomarker researchers who work with the Olink proteomics so that together we can have larger data sets and more reliable data. Well, there are several challenges in the development of any biomarkers and bringing them to clinic. For example, also for Alzheimer's disease, but for other neurological diseases. But for neurological diseases, it's a key challenge that we need cerebrospinal fluid. And it's a very powerful fluid because in Alzheimer's disease, we also know from our initial analysis using the Olink database platform that we see more signal in the CSF than in the blood if we have paired samples. So the CSF really is the matrix of choice for any neurological disease. By the way, we do not see that only in Alzheimer's disease. We also see it for frontotemporal dementia and also in multiple sclerosis. So for neurological diseases, we need to start or it's optimal to start with the cerebrospinal fluid as a matrix of choice. But that's also a precious fluid, so it's very important to collaborate to have access to larger cohorts and have more reliable data. There are, of course, other challenges, like in other diseases. For Alzheimer's disease, clinical implementation of novel diagnostic tests it's also very relevant that you have some some impact or actionability. So what does the result mean? Can you offer a treatment after that or for a patient? So there are several challenges, of course, as with other diseases. But yeah, I delineated already the uniqueness that in neurological diseases we like to have a cerebrospinal fluid. And Coral addresses these challenges because we collaborate, we merge data sets, or we take advantage of data sets. We do common studies using this cerebrospinal fluid. And also often the case that information that we obtain from in our studies in Alzheimer's disease can also inform the studies that we do for other types of dementia or for multiple sclerosis. So yeah, by collaborating, we will all learn from each other, and hopefully, or that should surely lead to some biomarkers that we can use in practice. I can give one example of a cross-disease study. It's not completely cross-disease, but first we did a very large analysis in Alzheimer's disease, CSF. And that was about 1,000 individuals of Alzheimer's, frontotemporal dementia, and Lewy body dementia. And but next, we continued a few years later with a study in familial Alzheimer's disease, and it was a small study. Only 22 patients were included, but we had very significant results. And by being able to relate it to our earlier data set, we observed that there was a very large overlap in the top 27 candidates. So by showing, having access to that earlier data set, and by being able to make a direct comparison, we could validate our data immediately. So that was very helpful. And we also studied other dementias, but that was also our primary goal. So I would not call that a cross-disease study. I know one. We also studied the Olink proteomics in multiple sclerosis with a different question to predict prognosis, disease progression in MS. So different question, different disease. But by the learnings that we obtained in the Alzheimer's analysis and studies, we could use that learnings and the techniques because for the data analysis for the multiple sclerosis patients. And in fact, we worked quite a long time on that, but we developed an automated or kind of script for data cleaning based on our earlier data sets, and now we can apply that. So we worked about 2 years on that script, but now this helps us to perform the data cleaning and also to generate the first graphs of disease differences for any novel data set for neurological diseases within on average 1 day only. So that's, I think, a very good output. Data quality is very important, and standardization also. So by doing the same type of analysis on Olink, that's already point 1. But the second issue, that's what we are working on now, is to include the same kinds of quality controls to be able to bridge between different analyses. So that will be a set of Coral quality controls that people can run along with their study, but that allows you to make an even more direct comparison between earlier and other data sets that use the same bridging samples. And of course, data quality also encompasses the data that are available in the databases. But of course, that's also the task and responsibility of the partners. So the consortium has now reached a nice stable core of people who are always there, very active members. And it's also time to grow further. And there are more people who are interested and work with the Olink tools, and they are open to join the Coral Consortium. It's an open consortium. And they can learn from what's available there, the expertise that's already available, and also learn by the interactions, and that's of mutual benefit. And I think because we have a nice core of experts, it's very interesting. It can be very interesting for novel people to join and leverage the learnings that we already have. And that will, I expect it to expand further the more corals being used. And I put some emphasis on the analysis of cerebrospinal fluid so far. But of course, we're also measuring blood biomarkers, especially because blood is more accessible. So it's always nice to have some CSF to start with, but we also observed very interesting signals also using such a proteomics technology when we analyze the blood, so even for neurological diseases. Yeah, that's a good question. What can you do? People can contact us, so either via Olink representatives, but also I do think that via our website there is an email that people can use. They can also directly contact me, so they can get in touch easily. There are multiple ways.
Well, I think this AAIC 2023 can be a very exciting meeting now to see the results also first of some of the clinical trials that are giving very promising results, and also to see in the field of blood-based biomarkers how are we advancing on at this moment, and which steps are taken forward in order to see the blood-based biomarkers implemented in clinical trials and clinical settings. So I hope to see a lot of that. And regarding the second question about the difference with other conference, I think the AAIC, it's a kind of challenging question to answer because I noticed that it changed over the years. It used to be a very clinical conference, really focused on Alzheimer's disease, and it's a little bit spreading now to also try to understand and gather the molecular pathways and a little bit more basic science. So it's changing a bit, and it's mixing like also another important conference like ADPD, that it used to be more neuroscience, basic neuroscience, focused on not only on Alzheimer's, but also in Parkinson's disease and other dementia. So they are intermingled a little bit one each other because the other is becoming more clinical also. So both are very, very interesting, but I think they both should keep their fields. Well, regarding detection of Alzheimer's disease, I think we have lived a revolution in the last years, and now we are really, really able to detect people developing Alzheimer's disease-related pathology before the appearance of cognitive symptoms. And this is, and we are able to do it just with a simple blood test. So this is an incredible advancement for the field. Now the challenge is like, okay, we did the work at the research settings. How can we translate it into the clinic? How can translate it so patients can benefit for that? And that's what we are all working on now, trying to move this towards the clinic. And also that means also towards the clinical trials, because that will allow us to select better the patients for clinical trials and to have other outcome measures besides clinical settings that will come. And hopefully that promotes also the development of effective treatments for Alzheimer's disease. For developing strategies for effective treatments also, what we need is the reality is that we really completely still do not understand the etiology of Alzheimer's disease. We know that amyloid beta and phosphorylated tau plays a major role in the disease, but we still don't really understand what is the cause of this disorder. And it's quite challenging because it's a silent disease, and we know that it takes 20 years to develop. So trying to understand what is happening at which stage and what is causative and what is a consequence of the disease process is a real challenge that we have in front. Yeah, well, a technological advancement. I think we are also living a technological revolution. No, some technologies that allow us to measure proteins in very, very low concentration in blood and very accurately are playing a major role because we can detect proteins that we were not able to do so before. But I think at the end is the development that allow us to really make omics a reality. And I think that is helping a lot because we can really analyze a huge set of biological process in one unique sample at different scales, from the proteome level, from the metabolomics, lipidomics, genome. All these technologies are going to play a pivotal role, not so much alone, but in the integration of both of them, because Alzheimer's disease is a highly complex disorder. So I really hope that by integrating these different technologies, we can really understand what's happening on and trying to get rid of all the noise, biological noise, let's say, or all the comorbidities that goes along with aging and that probably makes it difficult to understand what is really specific for the disease. Maybe we can do it. So this brings also to a next challenge, because if you are doing omics integration, you really need a power to do so. You really need large sample sizes, and that is a challenge not only for Alzheimer's disease but also for other neurodegenerative disorders that we also have to focus on those, not on other neurodegenerative dementias such as frontotemporal dementia or dementia with Lewy bodies. So we need also the development of very well characterized cohorts, phenotyped, that allow us to also to generate this omics data. Well, the SCALLOP CNS initiative is a working group that we have generated or started this year. It comes from the SCALLOP. SCALLOP is a consortium that is based on people or researchers that that especially work or are focused on the analysis of protein quantitative trait losses at any stage. A protein quantitative trait losses allow us to really see the association between how genetic variants influence protein levels. And this is very important because proteins are one of the latest scaffolds that produce biological function, involving biological function. So it can really tell you a lot of what is going on and how it's going on. So this in the SCALLOP really focus on neurology, on the central nervous system, is the analysis of pQTLs, really focused towards trying to understand better neurodegenerative disorders or disorders related with the central nervous system. Well, as I said before, I think one of the challenges is to really understand the cause. pQTL analysis can help in understanding what are the molecular pathways that are involved in the development of the disease, not so much that they play a role downstream, but really what can be driven in the pathways, the molecular pathways and the proteins involved in the development of in the development of this disease, of what is causing Alzheimer's disease. Hopefully, that's something that could be a thing. So in that sense, pQTL analysis can be very relevant within the field of Alzheimer's disease. And with the SCALLOP, the good thing of SCALLOP is that it's a consortium, so it brings together people that have used basically the same proteome array, which is Olink, and genome. So at the end, we have a large data set that comes together. So this power analysis that I was speaking before, that it could be a challenge, may be overcome thanks to this initiative, because we bring together people with different data sets in order to answer different questions. And indeed, we just started, I think, less than 6 months ago, and there are already 2 projects, 2 potential projects that are being initiated. And at the end, we aim to either replicate findings that someone has found. This is of paramount importance nowadays in the research field. If you find something, we need to validate it. We need to have independent cohorts for validation. Or maybe the other approach is to do meta-analysis, to already gather all the information that we have in different matrices, and with all this power, trying to find answer of what are the causative players, not only in Alzheimer's disease, but also in other types of dementias. Yeah, so as I said before, the protein quantitative test loci, it tells you how genetic variation alters protein levels. If we do that at the CSF, we have to think that it's a matrix, it's surrounding the brain, and therefore it's a mirror to what is happening to the pathological mechanisms that are happening in the brain in vivo, so in that moment of time. It's not post-mortem, it's really in vivo. So if we try to do pQTLs analysis on this matrix, which is kind of reproducing the changes that the brain undergoes during the pathological process, maybe we can try to understand how there's genetic variants that are related increase the risk of Alzheimer's disease. What are the biological mechanisms by which these variants produce the disease? And this is a major role, because then we are really unraveling which are the mechanisms and the biological pathways that are playing a relevant role in the disease, and might be novel potential therapeutic targets for treatment development. So I think it's very relevant. And also a very nice thing, I think, that since you are analyzing a lot of proteins, it can give you several proteins or a huge number of proteins that might be related to the disease development. And this can be investigated in follow-up step for drug repurposing opportunities. That means that drug that has been developed maybe for other disorders for other disorders that we know that also are involved in or increase the risk, like cardiovascular diseases or other type of medicines, might be also helpful for Alzheimer's disease. So I think pQTL analysis are just starting. It's a field that is starting to burst, but I think we will hear a lot in the following years, and I think they can really play a major role in the advancements in the research field. Well, I think that we are going to be able to do very interesting studies, especially not only in Alzheimer's disease, but in other neurodegenerative disorders that are not so common, that the prevalence is not so common. Mm-hmm. And here I would like to encourage people working in the FTD field, because sometimes there is genome associated data there. So frontotemporal dementia, because it's a really challenging disorder with highly heterogeneous. We know little about it. So we really need to work with data sets. We really need to work together, and SCALLOP can be a very good source to do so. Well, I would really invite everyone to join the group. It's really, really interesting. We're really interested in presentations. It's not only restricted, or we are not doing so far restricted to Olink. I mean, actually, one of the aims that we also want to encounter or to achieve is to really check different proteome platforms and what we find with different proteome platforms. So I invite everyone to join. So basically, just look at the webpage, SCALLOP. You just put SCALLOP in the internet, and there you will find all the information to contact the person, and they will probably forward the information to me. Or you can directly contact me. That's also fine.
You know, AAIC is pretty unique in the sense that you get basic science all the way through clinical development. And I think that's really important. And the Alzheimer's Association has really been the leader in that regard. So I'm always interested to hear a little bit of some of the basic science, the way that folks are using new techniques and technologies and understanding and mining data a little bit better. And also I'm interested in certainly the clinical trial outcomes. So as a company that's in clinical development, we're interested to see how other people are progressing, the types of things that they're doing, and how that may apply to us to ultimately benefit patients more broadly. Yeah, so that's a really loaded question with a lot of different answers. I can answer it from where I sit. I think the folks that are, the basic scientists or clinicians that are trying to understand the disease more broadly have a greater challenge. But for clinical development, what we're really interested is how do we select the right patients for us and when do we get them? So we target the immune system, and we really believe that Alzheimer's is quite a heterogeneous disease, and what you want to do is treat the patients that are most likely to benefit. So to us, the idea is quite simple. If you're targeting the immune system, the patient should have some evidence of inflammation. So what we are looking for is we're enriching our population to select the patients that have these biomarkers of inflammation. Where I think it gets challenging is which biomarkers do we use? And that's the sort of thing we're still learning. So we have biomarkers that we're using, and we're just started a phase 2 proof of concept study. But those biomarkers we expect will evolve over time, depending on what the data look like and what the best biomarkers are. So it's a really difficult and important question. And I think depending on where you sit, it has a lot of different answers. Yeah, so I think some of the interesting things, so I think there's two things, right? I lump them into two different biomarker categories. I think there's biofluids, and there's neuroimaging. Let me start with the neuroimaging. We're really big into diffusion MRI. We think there's a lot of information to be extracted from that, and some new techniques that I think will be really, really helpful and requires a lot of work and development. I think the blood and the CSF biomarkers are really traditional and very important. Mostly blood because it makes it a little more, the translational utility is pretty good. Platforms and techniques that allow us to collect as little blood as possible and measure a wide array of things is really, really important to the field. And I think one of the reasons we're having this conversation is we've actually used your platform in our phase 1, and we're going to be using it in our phase 2 studies. And we think that the use of yeast, the amount of blood necessary to do the studies is really critical for us. So I really think those are two areas in which we can continue to refine development that will make it easier for those of us that are wanting to work with patients and get these biomarkers. So this is really, let me just say, first and foremost, my bias is towards the immune system. So we need to keep that in mind. But I've been studying immune dysfunction, I like to call it, chronic inflammation, if you will, for almost 20 years now across a variety of different diseases. And what's very clear to me is that once the disease starts to develop, it really doesn't progress and develop to a large extent unless the immune system starts to kick in a certain way. So we really believe that inflammation and chronic inflammation in particular is a key element of disease development and progression. And any therapeutic approach will need to take that into account. So we think this is one area in which, from a monotherapy point of view, we can make a very large impact. Although we do believe ultimately that this is a heterogeneous disease, and any given patient will require a number of different therapies for ultimate success. So we believe it's critical, it's necessary, and we need to start targeting it. So I'm gonna answer that. So as I said a little earlier, I think it's very important. You know, blood is something that is routinely used and analyzed, and I think patients are, you know, reasonably comfortable with it. So the biofluid platform is extremely important. I think, you know, as time goes on and we learn more, it's hard to know what's going to be important, but certainly something will be important. I take the position where we need to see a clinical change and then sort of move it back to see what biomarkers are really relevant. Because at this point, truly, we're guessing. So a platform that's extremely flexible to grow as the field grows, to allow us to assess a lot of different things, I think is really critically important. Yeah, I think that's where it gets a little tricky. I think, to me, that where we're going to see the biggest benefit is working back from a clinical response to certain biomarkers, and I think that's going to be uh, um, drug target specific. So you may see with a neuroinflammatory strategy a different set of biomarkers that predict response, and this, which is the way I like to think about it. And you may see from an anti-amyloid or you know, metabolism therapeutics that you'll see a different biomarker. So I think it's critically important. I'm not a. I think it's a very large challenge to work from trying to take, you know, Alzheimer's as a disease and find the biomarkers. But again, the larger the platform, the more things that we can look at, I think is really important. And, you know, also to be able to look at protein changes in conjunction with some of these more quantitative as opposed to some of these qualitative changes that these large omics tend to give you, I think is important. So yeah, really important, and a lot of work ahead of us.